Hi, I'm Andy McMahon. I'm the Faculty Director for the Optical Imaging Facility here in the Eli and Edith Broad Center for Regenerative Medicine and Stem Cell Research. Optical imaging is critical in all our research, and we have a wonderful facility here. And I'm going to have Seth Ruffins, who is the manager of the center, introduce you to all the fabulous machines we have.
Hi, I'm Seth Ruffins, Director of the Optical Imaging Facility here at the USC Stem Cell Research Center. In our facility, we're equipped with 4 high-resolution confocal microscopes. 2 of them are Leica SP8 microscopes, and we have 2 Zeiss microscopes, an LSM 800 and LSM 780. In this room, we're equipped with an SP8 inverted microscope with a white light laser system. The beauty in the white light laser system is that we can excite fluorochromes from any wavelength between 460 and 670 nanometers. So whatever fluorochromes you need, we can excite them. Also, the microscope has a stage-top incubation system for doing live imaging for long periods of time. We've been successful at recording up to 36 hours just on the stage-top incubation system. Our LSM 780 is also equipped with a live cell imaging chamber for long-term imaging. Downstairs on the second floor, we have an LSM 800, which is a kind of a workhorse microscope, which has been very popular. Also on the second floor, we have the second SP8 with a white light laser system and a multiphoton laser. The multiphoton laser allows us to image very deep and gently into specimens. That microscope has been configured to allow for deep tissue imaging and intravital imaging. Also, we have a slide scanner with a 100 slide capacity magazine so that we can image many slides very quickly. The beauty in a slide scanner is that it allows for an unbiased view of your experimental sections. Not only are we an imaging center, we also help with image analysis and visualization. The analysis and visualization tools that we have here are Amira, Imaris, and SciGlass, a virtual reality system for analyzing your data. Also in the core, with the help of engineering students, we built an optical projection tomographer. That instrument has been used for the analysis of branching patterns in developing lungs and kidneys. So come to the Optical Imaging Facility and see what we can do for you.